All right, so welcome everybody. Good morning here. Good afternoon in Austria. It's a pleasure to welcome today Dr. Bettina Sonntag for our weekly seminar series. I'm going to pass on to Professor Laura that's here to make the introduction.
So good morning, everybody. Good afternoon, Bettina and Natalia that are in Austria. It's a pleasure to have today Dr. Bettina Sonntag. I have known Bettina for a long time from protozoology meetings. Bettina is a professor at the Innsbruck University in Austria, Mondsee, right? And it's a researcher in the Department of Limnology. And today she's going to present us a little bit about ciliates that are very interesting organisms. As I told you last week at the end of the seminar. So Bettina, it's a pleasure to have you here. Thank you very much for agreeing to give this talk. And I'm sure everybody will enjoy to know more about ciliates.
Thank you very much, Laura, and thank you both, also Eduardo, for the kind invitation to have a talk about ciliates, my favorites, and to show a little bit why they are key players in aquatic environments. So when you have a look into a drop of water and into the microscope, you see something like this, and you see many different organisms, interaction, and yeah, it looks very chaotic and diverse. But of course, we have here very nice relationships among all these organisms. Some are predators, some are prey, and this is called more or less the microbial food web or aquatic food web that you find in a small drop of water, for example, in 1 milliliter. And what is really astonishing here is that you have these incredible numbers of organisms that are found in such a drop of water. You find many different kinds of bacteria, photoautotrophic bacteria, cyanobacteria. Heterotrophic bacteria, you find many protists such as algae or phytoplankton. You find, of course, ciliates and also different flagellates and amoeba and heliozoans. And of course, you have a lot of viruses there. So in this tiny drop of water, you have an incredible amount of organisms that are constantly interacting. And so if you take a glass of water, for example, you can imagine that only from the number of organisms you find there and also viruses, you have numbers like we have humans on Earth, so several billions of organisms just in a glass of water. And each one of them is, of course, very important for this microbial food web, but also, of course, for the ecosystem. And here I would like to show you a little bit about the ecological diversity and why ciliates particularly are so diverse. Because you have, for example, in an aquatic water body, you have different habitats, such as like neuston, plankton, biofilm, or benton. And for example, here at the water surface, you can see that many microbes, also including ciliates, are living there because you have this high tension at the water surface, and this enables different microbes to crawl there, or even snails. Maybe you know the snails that are grazing on the water surface on bacterial biofilms and so on. And here we have then ciliates, for example, that are more flat, and they have specialized cilia or cirri. These are bundles of cilia that enable the ciliates to crawl on surfaces. Or you have others that are attached, and they filter food into their cell mouth. So, and in the plankton, for example, you have other ciliates that are more adapted to swimming or jumping. Some of them are have adaptations like longer cilia, and they can jump away when a predator approaches and so on, and some are simply swimming slowly. Then in the biofilm, you have again others, or also again such that you find at the water surface that are more crawling and that are grazing on the biofilms, or some are again attached to plants and stones or whatever is there. And finally, in the sediment, you find more flexible forms that are able to crawl in the sediment and search for prey, or that again sit on the surface. So simply when you look at all these different kind of habitats, you can see that you need special adaptation to this environment, and ciliates in particular are adapted to this, and this also causes this enormous high diversity of ciliates. Here you have some images from planktonic ciliates, and you can see just at one glance that they are highly diverse. They, for example, live in symbiosis with algae, or they build nice houses, or they are more floating forms or more hunting forms, or yeah, you see there is a really high diversity. But why should we study ciliates? Of course, they are really nice and look nice, and but they are, of course, key organisms in aquatic environments such as lakes and oceans, ponds, everywhere where is water, also in the soil, for example, you find ciliates. And in the plankton, for example, they belong to the most important consumers of algal biomass, and they are links in the trophic transfer to lower levels, to higher levels. And huge ciliates also are able to ingest, for example, rotifers or also larger metazoans, as soon as they still fit into this one cell of the ciliate. Then ciliates are very good indicator organisms, and this is because they have short generation times. So what does this mean? That ciliates can respond individually directly to impacts in an aquatic environment. This means when, for example, you have extreme weather events where you either can have heavy rain, then the whole community of microbes adapts to the situation, that maybe there is less food or there is a diluted environment, or you have some toxins that come into the river, for example, and then also some may tolerate them better, some others not. And because of all these short generation times, ciliates are excellent model organisms, and you can address your ecological or ecophysiological questions. So ciliates are really key organisms and very important in lake ecosystems or in aquatic ecosystems. Ciliates also have many different lifestyles. So here you have several images of ciliates, and those circled in blue, they are all of the same genus, which is Vorticella. And you see they look similar, but it is important that you identify the species here, because otherwise you don't see their ecological role in the aquatic environment. For example, if we have a look on this one, on the upper left one, then we see that this one is attached to a diatom colony, and they are regularly found in the lake plankton. Also in the lake plankton, you find others that are attached to zooplankton organisms. And all these relationships are very species specific. Then you have another Vorticella floating in the plankton, in the pelagic, and then you have another one that lives in symbiosis with algal endosymbions, for example. So you have four different species of Vorticella. And this is why it's important that you also identify the species, not only the genus. And for this, you need currently mainly microscopy. Then when it comes to food acquisition, ciliates are heterotrophic. Most of them are heterotrophic, and you can even see exactly what they were feeding on. So you can identify the food even also to the species level and identify this very specific relationship and the ecological role why a ciliate or what a certain ciliate species is doing in the lake or what it is feeding on or also who feeds on it. And then we have another very nice mutualistic relationship, so symbiosis kind of, where we find ciliates that live in symbiosis with algal endosymbionts. And in this case, the algae, they live on their own in the ciliate. They divide there and they also get shelter from predation or from so-called chlorella viruses. I will come to this back later. And these algal endosymbionts live on their own in the host. Then you have a second option of this so-called mixotrophy, because on the one hand, the ciliates feed on external food, and on the other hand, they receive photosynthetic nutrients from the algae. So this is a mixed nutrition, and it's called mixotrophy. And on the right side, you see another kind of mixotrophy that we also find in freshwater, but also in the marine environments. And you see these brownish spheres or plates inside, and these are chloroplasts of algae that the ciliate was feeding on. So the ciliate feeds on the algae and digests everything except for the chloroplast. So the ciliate can use the chloroplasts for photosynthesis, and these stolen chloroplasts are called kleptoplasts because of because it stole it from the algae, more or less. So we see that it's really important that we take a really close look onto the ciliates to identify their important role in the water body. Here again, this is just to show you that we have the same genus and different nutrition modes, because here we have algae, also here, and on the right-hand side we have purely heterotrophic species. So it's really important not to only identify the genus, but also the species. Yeah, and also just again, some few words on these mixotrophic species. When we were investigating lake samples, we usually go for a whole year annual cycles and see which species are there. And then we investigate the species richness and put it into relationship to environmental parameters or also to food and predators. And here, for example, usually in autumn, in the European autumn, you find a peak of mixotrophic ciliates in the water surface, and they make up more than 50% of the total abundance of the ciliates at some time. So this is a very important group in the lake. When we come to the ciliate communities and how they are distributed or structured along the water column, then we see again here at the lake surface that we have mixotrophic species, but also many other heterotrophic species that primarily feed on algae, because algae need sunlight, photosynthetically active radiation, and this is why there are a lot of algae and there are a lot of ciliate predators on the algae. The more down we go in the lake, we have strong abiotic gradients, because from what is produced in the upper part in the lake, when it's dying and sinking down more towards the lake bottom, bacteria come and they consume this material, and also down there we have then a complete different ciliate community that is more adapted to bacterivorous feeding style, and also they do not need any sunlight, because down there in these deeper lakes that are also typical for our region, so this means the lakes are around 50, 100, 200 meters deep, and then you don't have any sunlight and you have these heterotrophic ciliate communities down there. And depending on how high the consumption is, the more or less oxygen you also have there. And again, the communities are well adapted there. Then on the right-hand side, we have an alpine lake. And here a little bit more in detail, we have many of these small lakes. They are not larger than 1 hectare, so really small, and usually very shallow, so not more than 10 meters depth or even less. And they are located in a region high up in the mountains where you don't have trees and you don't have an, uh, an earth cover around the lake anymore, so you don't have a lot of nutrients coming into the lake. And usually these lakes are very clear, so they are very transparent. Also specifically for ultraviolet radiation. And this ultraviolet radiation also penetrates down to the lake bottom, even the harmful ultraviolet B radiation. So if you are a ciliate and you live in this lake, of course you have to adapt somehow. And it was quite interesting when I was working on these lakes earlier, because we found a handful of ciliates in such lakes. The ciliate communities were very stable over more than a decade. And for example, in this lake, we found only 3 ciliate species in summer. And the distribution was very typical. It was always one species that was thriving through the whole water column. And this species lives in symbiosis with algae. And these algal endosymbionts, we could prove that they produced some ultraviolet sunscreen compounds. And this was the reason why this ciliate could stay more or less everywhere in the water column. Then we had another ciliate that was very UV sensitive, and with this we tested also experimentally, and this is also a very food dependent ciliate, and this is why we found this ciliate only close to the lake bottom, because there was a lot of food available and they were sensitive, so they just stayed there and enjoyed the huge amount of algae. And then we had another ciliate species that was only and always found at the lake water surface. And because algae are able to produce these ultraviolet sunscreens, our assumption was, we didn't test it, that this ciliate was feeding on the algae, and because of the accumulation of the sunscreens, the ciliate was able to always stay here at the water surface. So we have very different niches, and each one of them has a certain adaptation to the environment. Yeah, this is just again an overview. Here you see also the ultraviolet radiation really from top down to the lake bottom, and that really also the shortest wavelengths are going down to the bottom. Here you see this lake. It's a very small lake in the Tyrolean Alps. Yeah, and it's very transparent to ultraviolet radiation, which is a key environmental factor for all the organisms living there. Usually you have low temperatures, less food available, and a thick ice cover for not anymore 9 months per year. All this information that I showed you so far is based on microscopy. And each one who knows, who is working with ciliates, of course, knows these blue books, despite 4 of them are in German, but there is a general key also guiding everyone to the description. So in these books, and they were compiled by Wilhelm Feusner, who was doing a lot of ciliate taxonomy, and he compiled these blue book. In these blue books, he compiled everything that was known about freshwater species, and he gave also a user-friendly key. So here you can see how these keys look like. So you have the ciliate group, and then you can just identify the one or the other species. What he also presented there was the ciliate communities. So if you find most of these ciliates that are related to a community in an environment, you can then relate it to a certain water body or soil or whatever, or even activated sludge. And for each of those species, a huge compilation of data is available in these blue books. And this is why these are really nice compilations for each ciliate species. Now we come a little bit more into sequencing, and we are a kind of back at the start now because taxon-specific sequences are not yet generated for the majority of the described ciliates. So this means when you do sequencing, you cannot retrieve this information because you don't get the information when you sequence your species only. At the moment, when you sequence a ciliate and you don't know which one it is, you didn't identify it at the microscope, you end up with a tree, with a phylogenetic tree like this, without any name. You might get an information that it's an environmental sequence, or if you're lucky, it tells you it's a ciliate. But this is a bit of a problem because at the moment, or because ciliates are, of course, very tiny, and all microbes are tiny. And microscopy is not so common anymore, because if you sequence an elephant, for example, then you know, okay, this is the elephant, and you can give the name to your sequence. But if you sequence a microbe, then this becomes a little bit more difficult. And so we are trying to fill up the databases and are investigating important ciliates in lakes. And one of them was Urodricha. So Urodricha is an important ciliate genus. There are about 40 described species, and you can also see here in this Oligodrichidaea community, you see that Urodricha species are also participating in this community. These Urodricha species are efficient heterotrophic algal feeders, and they are dominant in spring in temperate lakes. They graze down the phytoplankton bloom with other ciliates and rotifers. So in spring, we usually have these blooms of phytoplankton when temperatures rise, and then, of course, many predators come and just graze the blooms down. And for the larger species of the Urodricha genus, there is a key available, but not for the tiny ones. But there are several of them. Nobody likes them because they are extremely small. They can have only 10, 15 microns, so very hard to identify or to see any taxonomic details. And they are extremely fast. You simply miss them when you're not an experienced taxonomist. I like them a lot, but others not. Yeah, and so we did some with a master student. We did some cloning. We cultured several morphospecies from different populations, and we investigated the morphometrics, which means we investigated the cell itself at the microscope, and we sequenced the nuclear ribosomal operon. And finally, we also did some experimental approaches. So we did a kind of integrative taxonomy with morphology and also molecular sequencing. And wherever we do molecular sequencing, of course, we end up with cryptic species among the populations that we are sampling. So you get more work to do than you wanted to get done, but this is, of course, interesting. Yeah, and finally, we have a tree, we have the morphology, and we have also the names. So when you go back to Feusner's books and you have a name there, then now you also can retrieve a sequence. The same we did for another important ciliate genus, which is Colaps. In principle, it looks the same as Urotricha, but just with a calcareous armor. And they are, of course, closely related. Also very common, many known species. And usually there were 3 common morphotypes. We had 2 subspecies, one heterotrophic one green one, and there was another green species described. So from morphology, you could identify those 3 species and discriminate them somehow. But after having a look into samples from our lake just in front of our institute, I saw that there was a species that was not matching one of them. It was somehow in between. And this is why we did a more detailed study and found that we have 2 genetic groups again, that we have one group that has algal endosymbionts or not. So this is facultative endosymbiosis. So green does not mean that the ciliate is really an endosymbiotic ciliate, but it can also live without the algae. And yeah, so we came back to one Colaps name for the facultatively green one. And then we found another group where we only had one strain, and this was yet not found with algal endosymbionts, but we do not know. Maybe they will appear one day. But here, these molecular data show that we have 2 cryptic species in principle and 2 genetic groups, and from morphology, you cannot discriminate them anymore. Just also some few words on the green algal endosymbionts, because also the green algal endosymbionts need to be identified. And not only the symbionts need to be identified, but also the specificity between the host and the symbiont has to be tested in experiments. If they keep the same, if they like other symbionts, and so on. And also the identification of the endosymbionts. Here I have 2 experts in my research group who only deal with those endosymbionts. So you need experts for any aspect of your ciliate. And then, of course, you also have to check if your symbiosis is obligate or facultative, or how the whole connection between those 2 protists is related. And so finally, we found that in Colaps viridis, this is Micractinium conductrix. This was also unknown. And also for another ciliate, we found that Micractinium conductrix can be one of the endosymbionts found. Here, I just would like to mention that we were investigating the V9 region of the SSU and also the V4 region in our ribosomal operon. And for the Urotricha species, we found that the V9 region was not diagnostic to discriminate the species, but the V4 region was. For collapse, which is very closely related, it was exactly the opposite. The V4 region was not diagnostic, but the V9 region was. And for some study we did just recently on some hypotric ciliates, also the cytochrome oxidase, which also seems to be a promising marker, did also not discriminate the species. So it's a bit difficult to find a nice barcode marker for ciliates. When you have your ciliate species, then you can calculate so-called co-occurrence networks. So this means you are searching for significant relationships to food, to predators, and how your ciliate is related to the others. We did this for several ciliates, and for example, for this keystone species also in lake, Halteria grandinella, very widespread ciliate, we found that in the cold season and in the warm season, we have different networks. So this means that this species is very variably or can take different food at different seasons according to the availability in this case. But if you interpret such co-occurrence networks, in this case, these are based on sequencing, on environmental sequencing, and where we just threw in our ciliate sequence and had a look with which other organisms are they significantly related. So we have here two lakes, and we sampled the same depth, and you can see for this Colips viridis, this green or facultatively green Colips, we find very different networks depending on the depths we sampled and also on the lakes, depending of course on the other organisms present in the same water sample. When you now assume or know that your Colips is algivorous or omnivorous, then such a relationship of the ciliate to different algae makes sense. But of course, you need to know a little bit more about these dinoflagellates and if they could be a nice food for the ciliate, because they could simply be too large to be ingested. And also for this Micractinium, as we saw earlier, this is an endosymbiont of Colips viridis. And this can never be outside of the ciliate. So this finding is not a food-predator relationship, but it's really a symbiotic relationship, because we found in the same lake so-called chlorella viruses. And chlorella viruses attack these endosymbionts as soon as they leave the host. So we assume that we can never find the endosymbiont outside of the host. It also always has to be inside. And so you have to work like a detective, and it's really interesting, but you need to know the nutrition mode of the ciliate and also everything that is related to your food item, for example, or to a predator. And of course, you need to know which defense options does a ciliate have. Can it escape, and how does it escape? And can the significant relationships you found really be significant or not? And after you found a nice relationships, you can go for experiments. Then you bring your organisms to the lab, cultivate them, and for example, here we tested appropriate food and cultivation medium, again for an Urotricha. And what we found here in the lower graph, only one algae was an appropriate food item for our ciliate, and this was the only one that was swimming. All the other ones, either they were too big, or they were in colonies, or they were simply not moving. They were not interesting for the ciliate. This ciliate obviously likes hunting, and this is why it needs prey that is moving. Experiments for proving your hypothesis you can also do with ultraviolet or solar radiation. You can place your ciliates in a well plate, and then you can also cut off UV wavelengths, for example, and see if they were deleterious to your ciliate, and of course you have a control and so on. But it's important that you really experimentally confirm your findings. What else we usually do is that we have a look onto the seasonal and spatial occurrence of a ciliate species. This means over 1 year, usually, we sample the lake in different depths and over the season. So we have for each of those depth and season one sample that we were investigating from morphology, and then you can see species-specific occurrence of the species. And this is, of course, nice because you can see a distribution pattern. When is the species highly abundant? And then you can relate it to the environmental parameters that you measured along with sampling. When we do this quantification from morphology, this is fine. So in the first row, you have morphological counts from these different Urotricha species, again, from one lake. But if you do some eDNA or metabarcoding study and try to find the same pattern, then this does not work. So you can see these patterns are not matching. So you can currently not relate your molecular metabarcoding samples to the morphological counts that you find in the lake. Why? We did a study some time ago where we compared the morphotype abundance and the read abundance of the OTUs that we found in a metabarcoding sample. And why do we have such large discrepancies between those two kind of abundances? Because when you count ciliates in a water sample, then you can relate it to the volume and you can calculate how many are there. When it comes to read abundance, then you need to consider several things. You have, for example, a huge ciliate, and a huge ciliate means you have usually a huge macronucleus. Ciliates usually have two kind of nuclei, a macronucleus and a micronucleus. And in the macronucleus, of course, you find lots of DNA. So when you compare this to a tiny Urotricha species where you have only one macronucleus, then there is a huge discrepancy alone due to the size of the macronucleus. So this is really a huge discrepancy. So if you now trace by molecular methods one cell of the small one and one cell of the big one, then of course you have a much higher read abundance for the larger cell than for the smaller cell. And also the abundance in the lake is very different. So you have usually of the larger ones, you have less than of the smaller ones. So you have a huge discrepancy when it comes to abundance. And this is still a problem that we are also trying to solve. So there is a PhD student in the lab of Thorsten Stöck in Kaiserslautern, and she was working with digital PCR and now got some idea how this could work with Urotricha species. This publication is just accepted. And yeah, but in principle, you need to do this for every ciliate species. And even then, it's difficult to know how many DNA copies do you have in your ciliate because you don't know in which biological state the ciliate is when you when you retrieve the sample from the lake. So maybe it's a dividing one, maybe it's a conjugating one, or whatever. So this is really difficult, and this is one reason why these abundances don't match. When you have a sequence, then of course you can do nice things, and you can put your sequence into large databases and see if where your ciliate appears. And this is what we did with our ciliates, with the Urodricha species. We had large databases, metabarcoding databases from the whole world, and we saw where we found our ciliate. So this was a really nice approach that we are currently always using, because it's very interesting to see the biogeographic patterns of the ciliates when you have a sequence and a name. Yeah, and when you have a morphological investigation, then you can find ecologically important relationships, such as the one I will show you here. But if you have only sequences, then you would not find it. So we have a ciliate, this one, and this was found in the lake with a peak twice, here, and there was kind of leg face. I'm sure we would have found some explanation for this, but after looking into the microscope, because exactly there we took a sample, we saw that there was another ciliate parasiting this other ciliate, or the ciliate. And if you would have had only 2 sequences of ciliates from a metabarcoding study, then you would never ever have seen this nice relationship. So a look into the microscope is often still necessary. And here is just a short overview onto the life cycle of this Suctorian parasite and how this infected our hypotrichs. So you have we found swarmers, they attached on the ciliates, they embedded in the ciliate and so on, and they divided, they conjugated, they formed cyst, and you really saw a nice kind of cycle. And after the disappearance of the Suctorian parasite, the population of the hypotrich established again. So finally, my vision as an aquatic ecologist is that you take a drop of water, you put it on a sequencing device, and the species inventory pops up because all the sequences are already there. I know this is possible in principle already, but the sequences with the names are missing. And then immediately the species inventory pops up, the water quality is assessed, and also AI or somebody generates a summary on the background information, on feeding type symbionts, environmental preferences, movement, and so on. And maybe finally we end up with Ciliate Bible number 6, because 5 are already there. And you cannot see it properly, but I put some barcodes here. Maybe you can then just retrieve all the information and the barcode and all this huge literature archive that is already collected behind each ciliate species. But when you do not have a name to your ciliate, then this archive will not be opened, and you miss a lot of information that has already been collected and have to go back to start to do all the measurements and experiments. And I hope I could show you what you need to unravel ciliate out ecology. So you need the ciliate, its morphology, single cell sequencing. You can calculate the networks from metabarcoding samples. You can investigate the symbionts. Then you need limnological data, seasonal data. You need to know what is the food, how frequently are they appearing, in which area of the lake, and you can do experiments and whatever. And my very last point is, please do not forget also to communicate your science to the public. This is what we regularly do at our institute, that we have courses for children, for pupils, for schools, because this is really important that also the public understands what we are doing, why we are doing it, and why these tiny organisms are so important for our environments. So let me thank you. This is a view from our institute, and we are directly at the lake. So you are invited, of course, to come. And thanks a lot that you invited me to give this seminar talk. And yeah.
Thank you very much. That was excellent.
Thanks a
Everybody's
lot.
applauding here
Yeah,
digitally.
thank you, Bettina.
Oh,
It was
nice.
very, very good.
Thanks a lot.
I'm
So
sure
let me,
everybody here want to work with ciliates now. I will have
I
of
my
hope.
course,
full lab.
Yeah. They just have
of
to
course.
address you.
Yeah.
So I'll stop the recording here and then we can
Mhm.
move on to questions.